Hello, I'm Stephen Harrison from the Harvard Medical School, Children's Hospital Boston, and the Howard Hughes Medical Institute. Welcome to part 2 of this series on virus structures. This part is about viral membrane fusion, the process by which envelope viruses get into cells. As those of you who watched part 1 will know, envelope viruses, those with lipid bilayer membranes, acquire their membrane by budding out through the surface or into an internal compartment of the host cell. And likewise, they penetrate cells that they are about to infect by fusion, a reverse of the budding process, by fusion of viral and cellular membranes. Different viruses have different triggers or sensors, if you wish, to initiate the fusion process. Influenza virus, which enters through endosomes, depends on the low pH of the endosome to initiate fusion. Viruses such as HIV can fuse at the cell surface, and they depend on sensing the receptor, which triggers conformational changes of its own, and in the case of HIV, a co-receptor as well. What is membrane fusion? Membrane fusion is, in the simplest sense, making one bilayer out of two. But it's a relatively complicated process in practice, although it's thermodynamically downhill. That is, the fused structure is ultimately stabler than the two separate structures, but there's a substantial kinetic barrier, and it's overcoming that kinetic barrier that is the role of the viral fusion proteins or of cellular fusion proteins. So an intermediate in the fusion process is generally accepted to be a structure in which the apposed monolayers, the apposed leaflets of the two bilayers have merged, but not yet the distal ones. And that's called a hemifusion structure or a hemifusion stalk. And while there's still some debates about the detailed organization of the hemifusion intermediate, it's clear from a number of studies that that is an important step en route to fusion. Indeed, the barrier between two bilayers and the hemifusion structure is one of the major kinetic barriers in this process of fusion, and there is probably a kinetic barrier between hemifusion and the ultimate merging of the distal leaflets that lead to the formation of a fusion pore. In the case of viral proteins, there's a sequence of events that's reasonably stereotypical, it turns out, even though the machinery, the molecular machinery driving this series of events may look very different. That is, the fusion proteins of different viruses, although from the point of view of their protein architecture, may be very different. The underlying process that they catalyze, and there is a real sense in which this is a catalysis, since, as I said, it's thermodynamically downhill, but with a high kinetic barrier. The sequence of events that they catalyze is reasonably stereotypical in all cases. And so before events begin, the fusion protein is in some conformation, and this is a purely schematic representation, and there are two bilayers. The bilayer of the membrane in the cell, and the bilayer in the membrane of the virus, and the bilayer of the membrane of the cell to which the virus is attached. Some event, proton binding or receptor binding, induces or fixes a conformational change in the protein, in the fusion protein, that leads to the formation of an extended intermediate in which a hydrophobic element, either an N-terminal peptide or a loop in the middle of an extended part of the protein structure, interacts with the target cell membrane. And that extended intermediate, which is transient, then collapses into a structure that is ultimately a stable structure for the fusion protein and drags the two membranes together. As I suggested, there are probably kinetic barriers from the point of view of the lipid bilayer itself, both between the two bilayer state and the hemifusion state, and between the hemifusion state and the final formation of a fusion pore. And it is the role of the fusion protein to lower that kinetic barrier, as suggested by these dashed red lines. We'll talk almost entirely about the fusion protein of influenza virus, the so-called hemagglutinin. It's a member of a class of viral fusion proteins, all of which have the following properties in it, sometimes because they were the earliest ones characterized in molecular structural terms, have come to be called class 1 viral fusion proteins. These proteins are synthesized as a precursor, which is cleaved usually en route to the cell's surface by a protease in the late compartments of the secretory pathway, furin, for example, into an N-terminal element, which is usually a receptor binding domain. Some viruses have proteins like this, but have a separate receptor binding protein. And a fusion module, or the C-terminal half in general, which is anchored by a C-terminal transmembrane segment in the viral membrane. Examples of this sort of protein, they are all trimeric assemblies of this sort of organization, are influenza, HIV, and the filoviruses such as Ebola. In the case of influenza, where the protein hemagglutinin sticks off of the surface of the virus along with another enzyme, another protein, which is an enzyme, called the neuraminidase, and we won't talk about that today. The hemagglutinin is a trimeric structure, as I suggested, with three functions. It binds the virus to its receptor. The receptor is sialic acid on glycolipids or glycoproteins on the surface of the target cell. It has structures on the outside that can vary without compromising its two other essential functions, so that the virus can evolve to escape neutralization by the immune system of its hosts. And finally, it is, as I've suggested, the protein that catalyzes the membrane fusion process when suitably triggered by proton binding. So as I've said, it's synthesized as a precursor. This diagram is overly complicated, but all that matters for today is that at the N-terminus of the so-called HA2, the precursor is called HA0, and the two fragments are known as HA1, hemagglutinin 1, and HA2. At the N-terminus of HA2 is a hydrophobic peptide, exposed if you wish. It's actually not exposed in the structure, but made N-terminal rather than internal by the cleavage process that interacts with the target cell membrane and is known as the fusion peptide. And then there is a transmembrane segment very near the C-terminus that anchors the protein in the viral membrane. So the representation here shows you the overall structure of the hemagglutinin. This particular representation is based on X-ray crystallography and does not show the transmembrane segment or the very short segment of about 11 residues that extends into the interior of the virus particle, or before budding, into the cytosol of the cell. As you see, most of the HA1 part, which would be, let us say, red, and The HA2 part would be green of one of the subunits. Most of the HA1 part folds into a globular domain at the top of the molecule. It contains the site for binding sialic acid. HA2 forms a stalk that projects it outward from the surface of the virus. The sialic acid binding site here, there's one on each of the three subunits, faces outward. It's the one very conserved feature of an otherwise antigenically variable surface that the molecule presents to the outside world. Here's a slightly more readable representation both of the monomer on the left and of the trimeric spike-like hemagglutinin on the right. Let's look at the monomer. As I said, the HA1 part is largely out at the surface with its sialic acid binding site. The HA2 part forms the stalk of the molecule. The N-terminus of HA2, remember that's the fusion peptide, is here tucked in along the threefold axis of the trimer. And so the fusion peptide is hidden and can't interact with hydrophobic targets in the structure of the protein as we see it here. But as you'll see, once exposed to low pH, once protons bind, a major unfolding occurs that allows this fusion peptide to emerge and interact with a target membrane. So here's the low pH triggered conformational change. And one way of describing it from the point of view of the monomer is that the HA2 part turns itself inside out. That is, the part of HA2, and perhaps it's easier to see in this representation with colored segments, the part of HA2 that's on the outside in the trimer, which is red and then merging into blue, is on the inside after the conformational change, and the part that's on the inside, green and yellow, turns around and comes up the outside. This structure is most simply described as a trimer of hairpin conformations. There's a fair amount of twisting and turning at the turnaround of the hairpin, but fundamentally you can think of this as three polypeptide chains that begin up here with the purple arrow which represents the fusion peptide. It's not represented here since this is based on a crystal structure, comes down and then turns around and comes right back up to the transmembrane segment, which would follow the yellow arrow. So the hemagglutinin then undergoes two irreversible changes in the course of its maturation and exposure to low pH, because indeed the conformational change I just showed you is irreversible. If you then neutralize, you don't go backwards. And that's because of the first irreversible change, which is the cleavage of a peptide bond. That now means that the structure we see, which is very stable if you keep it at pH 7, soluble flu hemagglutinin can hang around for months or years stably in the laboratory. But if you expose it to low pH very, very rapidly, it rearranges as shown, and that rearrangement doesn't go backwards. And it doesn't go backwards because there's no way of reknitting that peptide bond, since this structure is actually not the lowest free energy state. It's just there's a very high barrier here that's lowered when protons bind. And it is that second change and the free energy recovered from that second conformational change that is coupled to the process of membrane fusion. And so the fusion mechanism can be thought of as cleaving the precursor or priming this fusion machinery, localizing the virus to to the cell by receptor binding, ultimately by uptake into the endosome. And the triggering of refolding, in the case of flu by low pH, in the case of other viruses, let us say by a receptor or co-receptor binding, that leads to this stereotypical sequence of events. Exposure of the fusion peptide, that's that extended intermediate, insertion of the fusion peptide into the target membrane, and a folding back of the protein that brings together the target and viral membranes. And it is that folding back that overcomes the first of the kinetic barriers. There is a substantial kinetic barrier to squeezing two membranes any closer together than about 10 or 15 angstroms. That is why liposomes, let's say, in solution are stable. Although once fused, they are even more stable. But a liposome preparation doesn't spontaneously fuse because of that kinetic barrier to bringing two bilayers close together. And it is that process that is at least one of the crucial ways in which these proteins facilitate membrane fusion. And they do so by recovering free energy in this fold-back process because the primed state is in one way or another metastable. So the fusion of membranes by influenza virus can be thought of then as a triggering process. We don't show actually the sialic acid attachment here, but a triggering process that leads to dissociation of the HA1 domains at the top. But there happens to be a disulfide bond down here that keeps HA1 from actually floating away. But some experiments done already 10 or 15 years ago, actually more than that, nearly 20 years ago now that I think of it, showed that if you knit the tops together, then this process can't occur. So we know that this dissociation of the tops from the stalk occurs. And that allows the stalk, the HA2 stalk, to unfold and refold, so to speak. That is, allows the fusion peptide to flip up, associate with the target bilayer, and then, along with the rest of the protein, collapse together to squeeze the two bilayers together, leading to membrane fusion. I said that the structure of the post-fusion conformation of, or the description of the post-fusion conformation of the flu hemagglutinin of HA2 corresponds to a trimer of hairpin-like structures. And it turns out that for large numbers of these so-called class 1 viral fusion proteins, that simple analogy is true. Indeed, in the case of HIV and SIV, the hairpin is particularly simple. It's just a helix coming down, a loop turning around, and a helix coming up. And so the membrane fusion process is nicely represented in this animation from Gail McGill, based on the structure of the post-fusion state of the HIV and SIV conformational proteins, which you can see going from the extended intermediate at the beginning to a fused state at the end. So we can then ask, in the case of flu hemagglutinin, which makes a post-fusion structure that's also a trimer of hairpins, although as it happens, as you saw, the outer layer isn't a simple alpha helix. The structure is a bit more complicated, but it's still fundamentally just a coming one way and going back the other way. How many trimers are needed to make such a fusion structure? And indeed, how long does the process take? And so in some experiments that our laboratory undertook with the collaboration of Antoine van Ooyen through the work of a graduate student named Dan Floyd, we sought to use contemporary techniques in single molecule fluorescence microscopy to try to carry out measurements of fusion looking at individual virus particles, because it was clear that the only way we could begin to answer the questions I was just raising about time scale and about numbers of hemagglutinins needed could only be answered in that way. And the experimental setup that Dan Floyd devised is shown schematically here. A lipid bilayer supported on a thin layer of a dextran polymer is doped with a bit of ganglioside, lipids that have sialic acid on their head group and therefore are receptors for flu hemagglutinin. An influenza virus that has been exposed to two different fluorescent dyes, that has taken up two different fluorescent dyes, is allowed to bind to this surface. The green dye is a hydrophobic dye that inserts into the membrane. The red dye is a more soluble dye that can be soaked into the virus particle and then the excess washed out and the virus used in the experiment before any of it leaks back out. And so those two dyes report, on the one hand, mixture of lipids in the two membranes, and hence the hemifusion step, and formation of an aqueous channel between the virus and the fluid, then the solute in the swollen dextran polymer layer that shows the formation of a full fusion pore. And finally, there's a fluorescein pH sensor to tell us in the bilayer, fluorescein is bleached when the pH drops below about pH 6, and that tells us when in the experiment that you're about to see, the pH in the region of the virus particle fell below a critical value. And so here's the kind of measurement that is done. And you'll see here the recording both of the signal from the pH sensor, the signal from the green dye that's in the bilayer, the hydrophobic dye, and the signal from the red fluorophore that is inside the virus particle. And what happens when the pH drops is that with a certain time delay, there is suddenly a rise and then a rapid fall of the hydrophobic of the fluorescence from the hydrophobic fluorophore. That's because there's enough of it in the membrane that the signal is quenched. This represents dequenching as the two membranes begin to merge, as the hemifusion event occurs, and then the fluorophore diffuses away in the target membrane. And then with a further time delay, there is mixing of the content of the virus with the aqueous substrate in the dextran layer underneath the bilayer, and one sees loss of fluorescence from the dye that was inside the virus particle as it diffuses away. And so if one does lots of these measurements, one can, and they can be done in parallel, because in a suitable microscope, as you see here, there are lots of particles in a field. Then one can get a histogram of the times to hemifusion, that is lipid mixing, and the times to pore formation. We can do this as a function of a variety of parameters, including the final pH of the buffer that was flowed into the little chamber in the microscope, and other parameters of the experiment. Analyzing this kind of experiment, in which, and I guess I should go back to explain that, as you see, hemifusion always involves a rise and then a fall. And when you have a kinetic event that has a delay of that kind, and so then if we're looking at the time to hemifusion, there is a certain delay that has a distribution from particle to particle that looks like this. then you know that there are multiple kinetic steps. Whereas if there's a simple single kinetic step, you would just see a single exponential decay, as you indeed do in the time, if you, on a particle by particle basis, plot the time between hemifusion and fusion. So to fit the kinetics of the hemifusion event, we chose a relatively simple kinetic scheme with two parameters, in which there might be n sequential steps, each with a similar rate-limiting steps, each with a similar rate constant k, or in independent parallel steps. And they turn out, under suitable conditions, have essentially the same sort of functional behavior. And as a result, if we fit these histograms, fit the histograms that I showed you in the previous slide, we find that the best fit involves an n of 3 and a rate constant appropriate for the times involved. These experiments were done at room temperature, of about 20 seconds or so as a kind of mean time to hemifusion under the conditions of this experiment. Whereas the pore-forming event was a single kinetic step from the hemifusion state to pore formation. Now, what's the interpretation of this sort of kinetic analysis? Well, as I said, there were several possibilities. One might be that there are n sequential steps, 3. Another might be that there are 3 parallel steps. By looking at the pH dependence, as indicated here, we found that n was essentially independent of the final pH. And it seemed to us unlikely that one could have n distinct sequential steps that would vary identically with pH, whereas the same is much more likely to be true of n parallel steps. And so we've interpreted n as representing the number of hemagglutinin molecules, the number of hemagglutinin trimers needed to form a successful fusion pore. That number, of course, might be 2 in some cases and 4 in others, but on average, over the large number of events analyzed, the number comes out to just about 3. In other words, that the free energy recovered from 3 hemagglutinin conformational changes appears to be sufficient to drive the process that I was showing you in the previous slide. So, which of the various steps in this sort of scheme are we looking at with that, is the rate limiting step in this sort of analysis? From the pH dependence, and I won't go into the details, we believe that there's an initial rapid equilibrium between a protonated and unprotonated state, but that as soon as this extended intermediate forms, then the process is essentially irreversible. And indeed, as a result of looking at some variants of the hemagglutinin, we're pretty convinced that it is this step that in the measurements I just showed you, we're looking at. So there happens to be a very conserved interaction just where the fusion peptide tucks in to the trimeric stalk. And mutations here, either in a completely conserved aspartic acid or a completely conserved glycine residue that stabilize the tucking in, mutations here accelerate fusion. And so we take that as evidence that it is this step that we're looking at. Now, in practice, on the surface of the virus, there are very tightly packed hemagglutinin molecules. There are 2 of them superposed on this electron micrograph. And so it is also plausible that 3 of these hemagglutinins clustered in one region might well be the minimum needed to catalyze this fusion process. Also, because of the tight packing, it's very unlikely that in the surface of the virus, the proteins can move around very much. And so again, the process is presumably carried out by a local set of interactions at an attachment point between the viral membrane and the cell surface. So these sorts of measurements obviously are just the beginning at trying to understand the details of this sort of process. But here from now just about 50 years ago is an electron micrograph of influenza virus in what would now be called an endosome, to indicate to you, to give you a bit of perspective and to suggest to you that from this sort of information, at a stage when one didn't even know what the molecules on the surface of the virus might be, we're now at a stage where at the level of dissecting the kinetics of the event and hence trying to understand sensitive points for neutralization by antibodies, for example, we can actually get at the molecular details of the process that would lead to the release of the nucleoproteins from inside the particle. You can actually see some of them in cross-section here probably, into the cytoplasm through fusion of the lipid bilayer of the virus with the lipid bilayer of the endosome. I've mentioned Dan Floyd and Antoine Fontoyen. I should mention also Tiana Ivanovic and John Scahill as collaborators in the measurements I've been showing you, illustrating how one can use structure and biophysical measurements to dissect the fusion mechanism. And I should add further credit to Gail McGill, whose animation of the HIV fusion mechanism is particularly helpful in trying to understand what we believe these fusion proteins are doing. Thank you very much.